Okay, can you see the Excel file? Okay, so I'll send you this Excel file later. So this is just what is it called? Dummy data to give you a chance at analyzing using the equations in the practical schedule to analyze the data. Okay, so these are the spectrophotometric measurements. So these are the ones. So when you're measuring chlorophyll A, you capture the chlorophyll A on filters. So you have to carry out a filtration step first. Then you place the filter into acetone. Okay, acetone. The acetone will extract the chlorophyll A from the phytoplankton. Then your acetone will be greenish in color. So overnight extraction, then you measure. There are two formulas to measuring it. One is a trichromatic, which means it uses 3 wavelengths, and one is a monochromatic, uses 1 wavelength. Okay, the one in your practical schedule is monochromatic. Okay, a single wavelength measurement. So here you see two measurements, one at 665 nanometer, one at 750 nanometer. Okay, although I said this is monochromatic, why is there two measurements here? Because only 665 nanometer measures the chlorophyll color. The 750 actually allows you to compensate for the suspended solids, for the particulates in the water. Okay, so the 665 is a color, so this is monochromatic. Okay, if it's trichromatic, you'll have an additional 2 wavelengths, excluding 750. Okay, so you have 3, altogether 4 wavelengths if you're using trichromatic. Okay, so this uses the spectrophotometric method. So spectrophotometric measures light. Okay, there's another method which uses spectrofluorometry. So that one measures fluorescence, because I've already mentioned to you earlier that primary producers autofluoresce, so their chlorophyll pigments can fluoresce. But you need to have a spectrofluorometer. Okay, so spectrofluorometer, not every lab has it, but spectrophotometer, most of the labs will have it. So it is easier to measure chlorophyll A using the spectrophotometric method. But spectrofluorometry is of course more accurate. So if you were to open up your practical schedule, you'll see that the formula is as follows. Okay, it's 26.7 6650 minus 665A times V over V times 1. Okay, what's the small v, what's the large V? The small v is the volume of the acetone. In this case, it is 8 mils. The large V is the volume of the seawater filtered. So that's from practical 1. The amount of filter volume filtered, which is 500 ml. Okay. So 665 O and A. What does O and A means? O is the original here, and A is the acidified. Okay. So what about 750? How do you use 750? So if you look here, this gives you. So it's 665A is absorbance 665A minus absorbance 665 750. So for all these values, for example, okay, so 665, so this is 665. So to get 665 O, you need to take this and minus this. So that gives you 665 O. Whereas for 665A, you need to take this and minus this. So from here you have your 665A, then you'll be able to measure this. So remember your small v equals 8 mils. So small v equals 8, and the large v equals 500. Okay, so it's 0.5. Because for the formula, we've already taken into account that the volume of acetone will be in mils, whereas the volume of the seawater filtered will be in liters. So it's 0.5 here. So use this formula, okay, and use this, the way to calculate 665O, 665A, and then calculate for the chlorophyll A, and also the paleopigments for group 1 to group 5, okay? Okay. Calculate for group 1 to group 5 your chlorophyll A in microgram per liter. What are paleopigments? So chlorophyll A shows the total chlorophyll pigments in your water, but some of the pigments, chlorophyll A pigments, are actually degraded. So if your phytoplankton is eaten up by zooplankton, or is your phytoplankton died, then the chlorophyll A will degrade slowly into paleopigments. Okay, so sometimes we also calculate the paleopigments, especially in waters like mangrove waters and polluted waters. In these waters, the concentration of paleopigments can be quite high and can affect your chlorophyll A readings. So it's good to know the concentration of paleopigments. Okay, but here, most important, get the chlorophyll A concentration out. And then these are the hemocytometer readings, the cells that you measured from practical 1. Okay, if you remembered, from here, measure. This is how you measure it. These are the algal cells that you observe in a hemocytometer and then converted to cells per liter. Okay. So use these readings for the number of cells that you observe in the hemocytometer and convert it to cells per liter. Okay, and then these are the measurements for the light dark oxygen method. So if you were to take a sample of the water, okay, incubate one set in light and one set in the dark, then incubate it for 6 hours and then measure again. Okay, if you get this kind of readings, can measure the uh, gross primary production, net primary production, and also community respiration rates. Okay. So if you remembered, for gross, for net primary production, it is this minus this. Okay, so the unit will be micromolar per 6 hours. Okay, so you can change it to micromolar per hour later. So for GPP, then for community respiration, that will be this minus this. Okay, so that will be your community respiration. So for your gross primary production, if you remembered, it is the sum of this and this. Okay, so that's your gross primary production. Okay, so I hope that you all will be able to write a report from this. Okay, show me how you analyze, obtain the chlorophyll A measurements, calculations, your hemocytometer, cells per liter, get the final unit to cells per liter. Okay, then your activity, whether it's a NPP, GPP. Then carry out the correlation analysis among all the variables that you have and discuss. Okay, discuss whether chlorophyll A is a good proxy for your phytoplankton cells. Discuss whether the chlorophyll A biomass that you observe is related to the primary production measurements. Okay, so you can carry out simple analysis and then write a short report on this. Okay, any questions? Any questions? Okay, short report. Results and discussion is enough. Okay, results and discussion. I want to see how you do your calculation, how you do your statistical analysis, and see whether your statistical analysis makes sense. Okay? There's no need to write intro, no need to write materials and methods. Keep it short. Yes, it's individual, but use all the data here. I want to, yeah, that's all. Any other questions? Okay, now let's look at another one. This is the practical 5. We'll talk about the submission later. Okay. Let's look at this. This is practical 5. So you were supposed to do this. So why this kind of experiment? This is to show you the mortality of bacteria in seawater. Okay, I've already, I mean, throughout the lecture, I've mentioned many times how fractionation is important in oceanography, in when you study microbiology in the oceans. We use fractionation to separate the predator from the prey, separate different fractions of organisms in the sea. For example, picoplankton, uh, nanoplankton, phytoplankton, microplankton, even macroplankton. So we separate them accordingly. Okay, but I've mentioned before that the separation is not absolute. There are always some outliers, but generally this is how we study, how we separate different organisms in the sea. Okay, so in environmental microbiology we use E. coli a lot, Escherichia coli. We use Escherichia coli a lot. We use Escherichia coli a lot because Escherichia coli is found to be a reliable indicator of fecal pollution in aquatic habitats. So the question is, if the presence of E. coli indicates that there is fecal pollution, but E. coli in the environment is also dying. It is dying because it can die because of seawater. It is not suitable to stay in the seawater. It cannot survive in the seawater, so it will die off. Or it can also die because it is eaten up by protozoa. So from your marine microbial food webs, you know that bacteria is eaten up by protozoa. So there is also so even E. coli in the aquatic environments are eaten up by protozoas. So if you have a sample of seawater, one is total seawater and one is fractionated seawater, filtered seawater, so less than 0.7 micron. So less than 0.7 micron means you're using a GFF filter. Okay, you're using a GFF filter. So one you don't filter, the other you filtered. Then you inoculate it with E. coli. Okay, you inoculate it with E. coli. This is a power 345. So please take note that this is from spread plating at 0.1 mil of inoculation. Okay, 0.1 mil of inoculation. So this is all done spread plating, 0.1 mil. Okay, so these are the counts you obtained. Then you incubate them, and after 24 hours, you obtain these results. Okay, then you will notice Okay, you will notice that of the counts decreased for the total seawater, but although the counts also decreased in the less than 0.7 micron fraction, the decrease is not at a high rate, as high a rate as the one in the total. So you will have two slopes. Okay, one where you measure. Okay, for example, for this, the count is 1500 CFU per mil, whereas for this the count is 1490. It's 1500 because it's 0.1 mil. Okay, so after 24 hours you get about 100 here, and here you get about 1500. Oh no, this is higher because this is power 3. So 1500 1500. Okay. Okay. So you have you have this kind of counts. So for total the difference is it decreased from 1500 to 100. So the decrease is from here to here. Okay. Whereas there it actually increases. So maybe it will be better if I do this so that you can see the change. Okay. So it actually increased here and it decreased here. So from here, what kind of discussion can you talk about? No, in the total seawater it decreased. Less than 0.7 micron, it increased a little bit. So why is there a difference here? Anyone? Why do you think there's a difference between the total fraction and the 0.7 fraction? Anyone? Okay, so in the less than 0.7 micron, there is no protozoa. Presumably there are no more predators. Here there are predators. That's why the grazing is there, and that's why your E. coli counts decreased. Here there's no grazing. That's why your E. coli didn't decrease. So from here, you can estimate the amount of grazing that has occurred, okay, between this and this. So from here, probably the amount of grazing will be okay, because this slope goes up, this slope will go down. So the difference between this slope versus this slope, this shows the CFU per mil per day. The amount of grazing that occurred, the amount of bacteria that's being eaten up. Okay? So for this, I also want you to write a report on this. Tabulate the data out. Okay? Then calculate the grazing rate for each group. Then you can do an average plus minus standard deviation. Okay, so what I then I also want you to answer this question. Okay, so for practical 3, include this question inside. Answer the advantage and disadvantage of chlorophyll A method to primary production biomass as opposed to counting phytoplankton. And for practical number 5, answer these 3 questions. The implication of E.coli mortality towards untreated sewage discharge. Then the difference observed between, why was there a difference observed? And then the effects of viral lysis towards this experimental setup. Okay, so answer 3 questions here. Question 4, 5, 6. And for this, answer question 3. Okay, so